Dr. Fiyazur Rahman. I have done my PhD in structural biology and my postdoc is in computational biology from this beautiful place, University of Birmingham. And nowadays you can see this is wholly covered into the snow. And of course winter is also here. So today we will run a marathon race about the drug designing. So I'll show you many ways that how you can design the drugs, how you can manipulate your certain compounds, which can be the synthetic compounds, they can be organic ones, inorganic ones, they can be the phytochemicals which you extract from the different phytoextracts. So there are a lot of things which you will be able to do and you will get a lot of ideas about these things. So I'll use, I think, hello? Yeah. Okay, I think you have seen this beautiful galaxy here of the stars. Many stars in the world, and you can see these stars when there is no any smoke on the sky, so as there is a chemical space. In chemical space, there are about 10 raised to power 60 chemicals available, and druggable out of them, they are only 10 raised to power 20, but still that's huge. Now we have to identify that which particular chemicals they are druggable, which can be used in the therapeutics. And then this chemical space, about 1 billion compounds with up to 13 heavy atoms, and there are 65 million known compounds which are available in the different databases. We are going to discuss few of them. A typical pharmaceutical compound collection, it may contain 1 to 5 million compounds. So that's quite huge if we want to use them for the therapeutic purpose, for the drug designing purpose. So what are the tools which we want to use for this purpose to identify the compounds which have the potential? And then if we talk about on the other side of the mirror, that is the biological space. I think you people are aware that there are about 30,000 total genes which are available in the human genome, which are expressing the proteins. And if we identify the druggable compounds, they are only 3,000, but still that number is huge because only existing drugs, they are based on 450 targets. So मैं हमेशा ये कहना चाहता हूँ कि there is a lot of room, there is a lot of space in this dimension. You can work on it. You can identify more and more targets. You can identify more and more chemicals which can be synthetic. You can synthesize them. You can use the inorganic one, or you can just explore the plants. Okay, so our goal, that is to reduce this particular chemical space. We have to target the disease. So this is also a flow of drug designing, that how you design a particular drug. So you have to identify the certain disease target. Either can be metabolic pathways, or either they are coming from certain disease expression as a disease expression marker. So you target proteins, you screen them, and you just try to bind them with the lead stain, and then you optimize the lead. We will discuss this process. So this is a normal drug discovery process, the conventional one, but I have merged it with the certain things, like when you are at the target identification, you use the certain bioinformatic tools, and then target validation, in which you use the druggability, the computational system biology. You identify the targets, and then you identify the hits. You identify the certain chemicals which are present in the nature. It can be fragment based screening, it can be structure based screening. हम इसे discuss करेंगे अभी कि ये होती क्या है. And then there is a hit to lead. Hit optimization is their preclinical trials, and then ultimately we go into the clinical tests. This is the process of the drug discovery, but this is not a limit. We have a lot of things which are related to it. We have the drug, we have the target, and we have the disease, which are related to all these domains: the network analysis, transcription signatures, protein comparisons, then binding site detections, docking. Docking is there, ligand similarity is there, and then data mining and machine learning. They have been incorporated into this process of the drug delivery. and it has totally revolutionized this drug delivery process. ये हम देखेंगे आखिरी जो कुछ मिनट्स है, मैं इसके ऊपर कुछ दूंगा कि कैसे एआई ने, आर्टिफिशियल इंटेलिजेंस ने और मशीन लर्निंग ने रेवोल्यूशनाइज कर दिया है ड्रग डिलीवरी के प्रोसेस को और इवन ड्रग डिस्कवरी को. ओके, आई विल से ओवर 100 डिसिप्लिन दे आर वर्किंग टुगेदर फॉर दिस ड्रग डिस्कवरी प्रोसेस. Okay, so my talk is usually for the students, so let's start with very basic things. So drugs is a small molecule which binds to certain targets. Okay, and what are the targets? Of course, these are the proteins. And drug discovery, that is the process by which the potential medicines they are identified. And then it involves a wide range of disciplines, mean many of them, over 100 as I said. Okay, so in this process, after drug designing, especially what you can do, you people can do as a student especially, you have to identify the biological targets. That's our first step. And that can be structural based or if any structure is not available. Because याद रखें कि जब भी आपने इन silico drug discovery पे काम करना है, आप चाहते हैं कि आप काम करें कोई ड्रग डेवलपमेंट के ऊपर, ड्रग डिजाइनिंग के ऊपर, या आप इन सिलिको स्टडीज कर रहे हैं, तो पहला आपका गोल होता है कि यू हैव टू आइडेंटिफाई द बायोलॉजिकल टारगेट्स. तो जब हम बायोलॉजिकल टारगेट्स की आइडेंटिफिकेशन करते हैं तो आइदर इट कैन बी स्ट्रक्चर बेस्ड और इफ स्ट्रक्चर इज नॉट अवेलेबल देन वी गो टुवर्ड्स द मॉडलिंग थिंग, वो हम खैर डिस्कस करेंगे. सो मेनली दिस इज द प्रोसेस, टारगेट आइडेंटिफिकेशन, then there is a visual screening process, and then these are the certain things which are related to visual screening, chemical synthetic process, screening, identification and clinical trials. I will emphasize in this talk on these two domains कि टारगेट की आइडेंटिफिकेशन कैसे करनी है, विजुअल स्क्रीनिंग कैसे करनी है. कुछ चीजों को मैं डिस्कस करूंगा क्यूएस से, और लेकिन बट मेन एम्फेसिस विल बी ऑन द टारगेट आइडेंटिफिकेशन and the visual screening process. Okay, target identification. Let's start from this. So, if the target is available, if no, we have no any target, what we will do then? Okay, then there is a process that we can do ab initio modeling. There are certain tools available for that. Either we can do the threading process, the fragment based modeling. There are a lot of tools available, few of them are here. Or we can do homology modeling process. Still, there are a lot of tools of homology modeling are present here. And then there is a discovery of AlphaFold 3. And believe me, the discovery of AlphaFold 3 has totally revolutionized the molecular biology, structural biology, drug designing process. It has totally revolutionized the thing. And you people must know that 2024 Nobel Prize that was given for the protein prediction process. And that prediction was based on the AlphaFold 3 discovery, that how it was made. So this AlphaFold 3 that works on that particular process, it works on the machine learning process. थोड़ा सा हम इसे डिस्कस करेंगे, but machine learning it involves the artificial intelligence, deep learning. You give the data to the computer learns from it, and then you ask the questions, and based on the learning of the computer, it answers you. In the same way, the protein are predicted. Okay. So second thing which is most famous, that is the homology modeling. Many people are doing that. So these are the steps. You select the template, and on the basis of template you build a model. That's quite easy and quite fair nowadays. If you have a sequence, you have got it properly from a database, you can make the model of it. And then either you can get three-dimensional structure if it's available from the protein data bank, or you can model it. Second thing is modeling. And after doing that, don't forget to check it. Don't forget to validate it. अक्सर जो स्टूडेंट मिस्टेक करते हैं homology modeling के अंदर वो ये होती है कि they don't validate it. वो उसे चेक नहीं करते कि आइदर आपका जो स्ट्रक्चर है जो आपने बनाया है by using certain software. देखिए, मॉडलिंग प्रोटीन इस चीज का नाम नहीं है कि you have the sequence, आपने एक सर्वर को दिया, उसने आपको मॉडल बना के दे दिया. No, this is wrong. आपने उसके बाद उस स्ट्रक्चर को चेक करना है कि either this is right or wrong. फिर आपने ये भी देखना है, देखिए प्रोटीन्स जो हैं जब हम उसको देखते हैं ना, हम उसको दो टाइप्स में डिवाइड करते हैं. एक स्ट्रक्चरल प्रोटीन होती है, अनस्ट्रक्चरल प्रोटीन होती हैं, और समटाइम्स देयर इज अ मिक्सचर. प्रोटीन्स डिसऑर्डर्ड भी होती हैं, ऑर्डर्ड भी होती हैं. तो अगर प्रोटीन आपने ऐसी चूज कर ली है विच इज डिसऑर्डर्ड वन, इट्स वेरी डिफिकल्ट टू मॉडल इट, बिकॉज आपको हर सर्वर एक डिफरेंट स्ट्रक्चर देगा. देन व्हाट यू विल डू, तो उसके लिए ये जरूरी है कि आपको एक नॉलेज हो प्रोटीन्स का, लूप्स का, या आप किसी ऐसे पर्सन से इन कांटेक्ट हो जो कि जानता हो कि आप उन अनस्ट्रक्चर्ड रीजन्स को या डिसऑर्डर रीजन को मॉडल कैसे करेंगे. Anyway, so these are lot of databases are available for the sequences, experimental structures, analysis, predictions, protein data bank that is very famous to get the structures. So once we got the protein structure, we have also a list of unending list of the servers and the softwares which are used for the protein prediction and for the validation of the proteins. And once we have done with the modeling thing, we got the structure. So this was our first step. Second step is to identify the binding sites in the proteins. हमें पता भी तो होना चाहिए कि इस प्रोटीन के अंदर अगर हम कोई कंपाउंड जो है उसकी स्क्रीनिंग करना चाह रहे हैं तो किस जगह पे करेंगे. If proteins it consist of means the thousands of the atoms, या उसके जो अमीनो एसिड्स हैं 200 300 हैं, तो where the certain ligand that will bind. So there are certain softwares like the Cas2b is there. There are a lot of software others which are involved into the prediction of the binding sites. So Cas2b and find site, they are the important one, and you can use these ones. Okay, the second step is the lead optimization. That how we can do the lead optimization, the lead compounds. Again, there are several approaches. So some approaches they lead the structure-based strategies in which you use the protein structure and you design the certain ligand on the basis of that particular protein structure. Other is that is called the ligand-based strategies in which you modify the ligand. If you are good in computer, you know how to use the software. If you are good in organic chemistry along with that, and you know कि किस साइड के ऊपर कौन से बाइंडिंग ग्रुप्स हैं, वो हाइड्रोजन बॉन्ड बना सकते हैं, वो कोई और वॉन्डर वार फोर्सेस क्रिएट कर सकते हैं, हाइड्रोफोबिक किस साइड पे, हाइड्रोफिलिक किस साइड पे है, then you will be able to design a very good ligand molecule. You can combine both of these. You can just get the protein structure, आपको एक टारगेट पता चल गया है, and then further then you can define कि आपका ligand जो है वो क्या होगा. Lot of databases again which are available, you can download. So these are the commercial databases in which you can get the commercial compounds. Means ChemSpider 67 million structures are available. You can download almost any structure which you want.